Hello everybody and welcome into our presentation on bioinformatics. Our group, it consists of myself, Tyler, we have Camelia, we have Patricia, Jose, and Falos.
So my part of the project is to talk about bioinformatics and what it is. So basically, bioinformatics is a blend of molecular biology and data science, and it deals with the application of computer methods to analyze large collections of biological data, such as genetic sequences, cell populations, or protein samples. The term bioinformatics was created by Ben Hesper and Pauline Hogweg to describe the study of informatic processes in biotic systems, and it found early use when the first biological sequence data began to be shared. And computer science, chemistry, biochemistry, biology, statistics, mathematics, and engineering all have to do with bioinformatics. So it is a highly integrative field involving many different types of specialists, including biologists, molecular life scientists, computer scientists, and mathematicians. Analysis of the genome sequence data, particularly the analysis of the Human Genome Project, is one of the main achievements of bioinformatics to date. Prospects in the field of bioinformatics include its future contribution to functional understanding of the human genome. Traditionally, bioinformatics was used to describe the science of storing and analyzing biomolecular sequence data, but today bioinformatics is a much broader discipline encompassing modeling and image analysis, in addition to the classical methods used for comparison of linear sequence or three-dimensional structures.
Hello guys, today I'm gonna talk about my group project, DNA sequencing. I'm gonna break it down to talk about the DNA and the sequencing. DNA sequencing is basically the process of determining the sequence of nucleid based on that or A, T, C, and G is a piece of DNA. So today, with the right equipment of material sequences, a short piece of DNA is relatively straightforward. Sequencing is the entire genome, which is all organ DNA. We might have complex text is required, breaking the DNA of the genome into many small pieces. Sequencing, those are pieces, and assembling the sequence into a long consensus. And then in my project, the DNA is cut into fragments around 3,000 nucleid, 3,000 small nucleid. And then thousands of those fragments or the sequence simultaneously. Several groups been working in the next generation sequencing and beyond. Several groups have been working in the third generation sequencing, where in a single very long DNA molecule is sequencing isn't on. The next step is how they working. CGTEA is a replicate product of serial reaction, and then they separate by the group of gel electrophoresis. So the DNA sequence is the process of determining the nucleic acid, the order of nucleic and DNA. That's all for today. My part was DNA sequencing.
After the different versions of the genome shotgun method, we get into the advanced study of genomics. So what is genomics? It is the study of the complete sets of genomes. The first targets of genomics for research were bacteria. In 1995, there was a major leap forward with the breakdown of the entire nucleotide sequence of the influenza genome. From there, the study progressed to baker's yeast and then to more complex organisms. The majority of the organisms that were sequenced have been nearly 4,000 bacterial species and nearly 200 different forms of archaea. One major quote from the text that was important to this topic can be found on page 230. The quote reads, "As the ultimate repository of the genetic information from which all of life's inherited characteristics develop, genomes hold the key to our genetic identity." This part is incredibly important for the fact that the study of genomics and genomes in general lead out to what we'll follow up in our next video, which gives us a little bit of insight into what that is. Alright, so up next we are going to go over what genomic sequencing is.
Genomic sequencing is a process for analyzing a sample of DNA taken from your blood. In the lab, technicians extract DNA and prepare it for sequencing. Within every normal cell are 23 pairs of chromosomes. Chromosomes are structures that house DNA. DNA is coiled into a shape called the double helix. The double helix can be unwound into a ladder shape. The ladder is made up of paired chemical letters called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G. In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA, one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care. Discuss the results of the sequencing of your DNA with your doctor and a genetic counselor. Genomic.
Alright, so the whole purpose of that is it gives us up to the whole genome shotgun method. So what this is it's the process that's used in order to create the DNA sequences mentioned in the video. So the first step is that they're going to chop the entire genome sequence into fragments with restriction enzymes. They then take those fragments and they're cloned and sequenced. Step 3 is they're going to add the computer to reassemble the short sequences into a singular continuous sequence for every chromosome. The DNA sequences are then deposited into the GenBank database. This database is one that contains more than 200 billion base pairs of DNA. Any sequence in the database can be retrieved and analyzed further for more information. Next up, we'll be going to the Human Genome Project, which will be mentioned up by Patricia.
Hi, my name is Patricia, and I am going to talk about the Human Genome Project. The Human Genome Project and how it began. The Human Genome Project began in 1990 as an effort by the government-funded researchers from 6 nations. This project has provided a wealth of data that may illuminate the genetic basis of what it means to be human. At the completion of this project, more than 99% of genome has been determined to 99% accuracy. What this means is that the genetic studies of human inheritance characteristic has been found to be 100% accurate. The Human Genome Project and what it is. The Human Genome Project is an international collaborative effort that sequences the DNA of the entire human genome. The human genome is a complete set of genetic information for humans. The Human Genome Project was a massive scientific initiative to determine the nucleotide sequence of all DNA in the human genome, to identify the location and sequence of every gene. Human gene consists of DNA, and most of it does not endure proteins. The human genome is made out of 3 billion base pairs of DNA. And what makes up the human genome is DNA. These DNA are consisted of genes, and that's what makes us unique. Now you're going to see a video that gives you a brief overview of what I just talked about and what I just read about the human genome.
Thanks for stopping by. Today we are talking about the Human Genome Project. Before we discuss the actual project, I want to define what a genome is. So a genome is the complete set of genes in an organism. In the Human Genome Project, that organism is, of course, the human. There are several scientific techniques that made it possible to start and complete the Human Genome Project. These include DNA sequencing, polymerase chain reaction, and electrophoresis. The Human Genome Project was essentially an international research project to map the entire human genome, every section of DNA in a human cell, which is a lot, 3 billion base pairs, in fact. And they've mapped all of them, which took 13 years to complete. The project determined that there are about 20,500 genes in the human genome, and it also discovered the order and location of these genes. It didn't, however, identify what each gene does, and that research is ongoing and there's still a lot to learn. It continues to be a benefit to society as a massive information source for researchers to draw on and to continue their advances in both science and medicine. I hope this brief explanation